So my name's Hamish Scott. I'm head of the Department of Genetics and Molecular Pathology at SA Pathology in Adelaide, Australia, and we're also part of an academic alliance called the Centre of Cancer Biology.
And I'm Chris Hahn, and I'm head of a molecular pathology research laboratory.
So we've been working on germline influences to hematological malignancies for a long time now. And as such, families come to us for diagnosis. In this case was an Adelaide family, a small nuclear family of just 3 individuals, 4 individuals with the dad, where the mum actually had neutropenia and thrombocytopenia, and then went on to develop acute myeloid leukemia and myelodysplastic syndrome and unfortunately pass away. So both of her sons, who are now in their 20s, actually also had thrombocytopenia and neutropenia. So they came to us diagnostically and they tested negatively for all the genes which were known to be involved in predisposition to platelet disorders and hematological malignancies. So we're fortunate that we could roll those patients and those families straight into our research programs, and one of the first things we noticed was that they contained a sequence variant in a transcription factor called ERG. Now that's significant for a number of reasons. Many of the other genes which are involved in platelet disorders and hematological malignancy predisposition transcription factors, such as GATA2 and RUNX1. ERG was actually on our suspicious list of genes for many reasons. And in fact, this nucleotide variation caused a missense mutation, which is actually in an amino acid which is essential for DNA binding. So it was in all 3 individuals who were affected with the thrombocytopenia and neutropenia. So we did what lots of people involved in genetics as opposed to hematology do at this stage, and we entered the data saying that we had a variant in ERG, the gene, and what the phenotype was, into essentially a matchmaking service online called Matchmaker Exchange. We also phoned lots of friends, and from that we got a cohort of patients where we believed that many of them actually had genetic lesions in the ERG gene, mainly focused in the DNA-binding domain of ERG, the ETS domain, which caused thrombocytopenia, neutropenia, and a predisposition to hematological malignancies. We don't think we've finished describing the number of diseases and the number of mutations which will be associated with this disease or these diseases so far. However, I will let my colleague Chris describe some of the functional assays which we've done, which I think give us great confidence that we have in fact described a new gene and disease entity.
So we went on to do functional assays on the variant that we found in our Adelaide family, our Australian family, and also on a whole range of variants from our overseas collaborators. Because ERG is a transcription factor, we did transactivation assays to start with, and we're able to show that variants within the ETS domain, the DNA binding domain of ERG, were loss of function. And we were able to show that these were due to loss of DNA binding and also due to loss of nuclear localization. So there were 2 mechanisms here leading to impacting on ERG's function. So these were in vitro assays, and we wanted to look at some more cellular in vivo type assays. So in fetal liver cells, overexpressing ERG leads to cytokine independent growth in tissue culture, but it also transplanting those cells into mice leads to development of an erythroleukemia. So we took fetal liver cells, transduced them with a virus overexpressing wild-type ERG, and also with a range of variants. And we're able to show that these while wild-type ERG could cause cytokine independence and erythroleukemia in mice, the variants from our family and also from our collaborators that impacted the ETS domain of ERG, they were loss of function. They were no longer able to perform those functions. So we believe we have a new entity here, a new disease where an ERG deficiency syndrome, and we would encourage germline testing for the variants in ERG.
So there's probably one thing that we forgot to mention, and that was that in all 3 Adelaide family members, the variant in the ERG gene was actually hidden when you analysed samples from blood and marrow by a process called somatic gene rescue, meaning that in blood cells, the mutant allele had been swapped out for the wild-type allele to a large extent. We're not sure how common this bone marrow deficiency, pancytopenia and hematological malignancy predisposition gene is going to be in the general population, but it is also possible it's more common than detected to date because of this process called somatic gene rescue.